Let's take a look at an eyeball. So here we have a sheep eyeball, which is going to be a fair bit smaller than ours, but all the pieces and parts are going to be exactly the same. If we look at the outside here, so we're looking at the front of the eyeball, we can see here the cornea. And that's going to be the outer surface of the eye, the first thing that light passes through on the way to the retina. And externally, it doesn't look very exciting. We just see white eyeball around the edges and then the darker middle here, where we would have the pupil and the iris would give us the iris, the colored part, and the pupil would be the hole in the iris, and that collectively gives us this dark section of the eye. We can look here at the eyelid. So here's some of the eyelid right here that would have gone all the way around the eye, and so when the eye would move around, it would rotate within that eyelid. If we go to the back side of the eye and take a look here, I've dissected away a little bit of the fat that's normally found here on the back side of the eye, and what we find here is the optic nerve. So that goes straight from the eyeball itself and plugs into the brain. And if you've watched the brain dissection video, you would know that the optic nerve plugs directly there into the brain, right up underneath the frontal lobe at the optic chiasma there. So the eyeball has a direct connection to the brain. It doesn't have to go through the spinal cord or anything else. So what we want to do now is we want to actually go inside this eyeball and take a look at the internal structures. This particular eyeball is a little bit low in pressure, so we'll see how we go here. Eyeballs are made out of very tough tissue that does not readily slice. And if you're ever dissecting an eyeball yourself in person, do be careful when you actually slice into it. If you've got too much pressure, the eyeball juices will squirt out on you. It's never happened to me, but I've seen it happen to other people, and it's quite entertaining if you're not the person getting squirted with eyeball juice. Making some progress here, but again, it's really sturdy stuff, not readily disassembled. Okay, here we've taken it apart enough. So now if we look at this eyeball from the front side, we can see a lighter area here in the middle of this dark area, and that was the pupil. If we look at it from the back side of the eyeball, just kind of turn this inside out here a little bit, we can see this lighter area here, that's the pupil, so there were no colored pigments present there. That was relatively clear tissue. And this dark ring around the edge here, that's the iris from the back side. And that was giving the eye its color. So if you happen to be brown-eyed or blue-eyed or green-eyed, the iris itself is what actually contains those colored pigments that would then give that eye that particular appearance of color. There are two different kinds of humor or liquid in the eye, and what we would find between the cornea and the lens would be called aqueous humor. Humor just refers to bodily fluids and liquids, so the aqueous humor in this case would be more of a watery stuff, and this watery material that's poured out on the dissecting tray here, that was the aqueous humor. And then on the back side of the lens, so between the lens and the rest of the eye, we would find vitreous humor. And if we look on the back side of the lens here, we can find this more jelly-like material. See if I can carefully pop the lens out here. Okay, here's the lens, and all this jelly-like stuff on the back side, that's the vitreous humor. And the job of the vitreous humor is to hold the lens up in place. So it's pressing on the lens to the front, it's pressing on the retina to the back and holding everything within its place in the eye. Because otherwise it's not really well anchored. Now, like we said, this marble-looking structure here was the lens. And that's the part of the eye that actually focuses light that comes through the cornea. And it focuses it through that vitreous humor onto the retina at the back of the eye. And it comes out very easily, but there are some muscles that do attach to that, pulling it to stretch it and help light focus. But then it hits the retina at the back, and right here we have the retina. And the retina, as you can see very easily, comes out of place. because we've removed the vitreous humor from in front of it that was holding it up in place. But all this brownish tissue right here was retina, and it was nicely, smoothly pressed to the back of the eye here. We can find that it's really only attached in one place. So if I pull the retina back here a little bit, we find that it's all coming together at one point right here. So that's the attachment point for the retina, and again, the only place where it's really firmly attached. The rest of it, again, just held in place with that vitreous humor. But where the retina attaches right here is called the optic disc. And if we go to the back side of the eye at that same place, we find the optic nerve coming out. So the optic disc is the connection between the optic nerve coming in from the back of the eye and the retina. And this introduces the blind spot. So in your reading assignment, you would have read about blind spots, and I'll talk about it a little bit in the lecture as well. But the blind spot basically is where the retina plugs into the optic disc. There are no photoreceptors there. So that means there's nothing to detect light at that particular spot. So at the optic disc is a spot at which the eye perceives no vision whatsoever. And you don't notice a blank spot in your vision unless something gets very, very close to your eye. So if you were to point your finger towards your eye, the closer it gets to your eye, the less of your finger you actually see, because the more of your finger is becoming centered on that blind spot. But the brain has a very sophisticated process called filling in, which basically copies imagery from around the edges of the blind spot and pastes it in, assuming what was around the edges is what was actually in the middle. And for relatively small objects, when they actually get very close to touching the eye, are not detected because there's nothing of them being copied from around the edges and pasted in. We also see here some iridescent shiny stuff. So it's a little bit of iridescent blues, greens, a little bit of shiny black going on back here at the back of the eye. And that is going to be called the fovea centralis. And what that does is it sort of reflects light. So when light comes in through the cornea, through the lens, and through the vitreous humor, hits the retina, and actually some of it passes through the retina, but it helps to reflect the light around and make better use of the light that does come in. That's especially useful in low light conditions. So if you're driving along the road at nighttime and you see glowing eyes of an animal in your headlights, the glowing part of the eye is the light bouncing off of that fovea centralis and back out of the eye, producing that glowing eye effect. And that probably helps you to avoid hitting a lot of animals if you see that glowing eye first. In the olden days, before modern camera technology, there was a real problem with red eye in pictures. And so when the flash would go off, it would bounce off of that fovea back out of the eyes and give a glowing in the dark eye appearance in a picture with flash photography. Modern camera technology has figured out how to cancel that out and not cause that problem. So the eye, from a structural standpoint, is pretty straightforward and simple. Not a whole lot of sophisticated structure to it, but if we look within the retina itself on a microscopic level, we would find those rods and cones, those photoreceptor cells, being actually quite complex. But from a dissection standpoint, not a whole lot to it. That pretty much wraps up the eyeball for us. Thanks for joining us. Have a great day.